What are gene therapy and cell therapy? Gene therapy is a process of replacing treatment using the healthy and compatible genes into the person who has any disease or disorder rather than using drugs. Cell therapy is a transplantation of cells in the body that have been damaged or deteriorate. It will be done by repairing the cells or tissues in the body using the new technological innovative and products that would help the treatment of variety of disease or any conditions. How does gene therapy works? Gene therapy is for replacement of wrong or malfunctioning genes to improve body's ability to fight diseases. For this to happen, scientists use some vehicle called vector, which is genetically engineered to deliver the genes. An example of that is virus. In recent research, they conducted adeno-associated virus as a sample that can carry therapeutic gene and live on its own. It was being tested in both in the body and out of the body. Inside the body it was being injected and directly go to the liver when it is into the bloodstream. As this virus used, the blood clotting factor in the liver caused the virus to treat hemophilia. While outside body used by taking the bone marrow and separates its immature cells where it will be added the genes and inject into the bloodstream of the patients. As the cell travel, they undergo mutation that multiplies rapidly and replace all defective cells. What are the strategies being used in gene therapy? Gene therapy is an experimental technique that uses genes to treat and prevent disease. There are different strategies used in gene therapy. Gene augmentation therapy is used to treat disease caused by mutation that stops a gene from producing a functioning product. It adds DNA containing a functional version of the lost gene back into the cell. The new gene produces a functioning product at a sufficient level to replace the protein in the protein that was originally missing, targeted killing the specific cells. This aims to insert DNA into diseased cell to die. It can be done through direct killing or indirect killing. Targeted mutation correction is the repair in genetic defect to restore functioning allele. Targeted inhibition of gene expression. The basis of this therapy is to eliminate the activity of gene that encourages growth of disease-related cells. What are the uses of gene therapy? It is a technique that modifies a person's gene to treat or cure disease. Gene therapy is responsible for replacing a mutated gene that causes disease with a healthy copy of the gene, inactivating or knocking out a mutated gene that is functioning improperly, and introducing a new gene into the body to help fight a disease. It replaces a faulty gene or adds a new gene in an attempt to cure disease or improve your body's ability to fight disease. Gene therapy holds promise for treating a wide range of diseases such as cancer, cystic fibrosis, heart disease, diabetes. What are the common types of cell therapy? The most common type of cell therapy is blood transfusion, and the transfusion of red blood cells, white blood cells, and the platelets from a donor. How are viruses used in gene therapy? Viruses are used in gene therapy as gene delivery vectors and as oncolytic viruses. Viruses as gene delivery vectors in order to deliver the genes to the cell. Only genetically engineered vectors such as viruses are used in gene therapy. This newly engineered virus protects the new gene it carries from degrading enzymes in the blood. The virus, being obligate intracellular organism, forces the cell to take up the new gene and goes into the nucleus. Once in the nucleus, or for some, once integrated into the chromosome, the new gene starts to perform its function to synthesize a new improved or functional protein. Can viral vectors make you sick? However, like all treatments or therapy, it has its risks. Therefore, anyone availing this therapy must discuss with their physician and other health advocates the pros and cons of gene therapy. When viral vectors are used to carry genes into the body, they might infect healthy cells as well as cancer cells. Another danger is that the new gene might insert in the wrong location in the DNA, possibly causing harmful mutation to the DNA or even cancer. Generally speaking, viral vectors have more favorable safety profile than many live attenuated virus vaccines and are more immunogenic than inactive killed virus.